Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from neuroscientists across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, and for more info on the talks covered in this seminar series and all things neuro related here at Cambridge, please follow us on Twitter at CamNeuro and follow the links below. Okay. So welcome everybody to the Cambridge Neuroscience Interdisciplinary Seminar Series. It's great to see so many of you here with us today, and we're delighted to welcome Professor Michael Coleman. Michael, as many of you will know, is the Van Geest Professor of Neuroscience at Cambridge. Previously held positions at Babraham in Cologne and in Oxford. We're now delighted to have him firmly fixed at Clinical Neurosciences. So his group identified the first protein known to delay degeneration of injured neurons. And I'm sure he will tell us all about that today and much more, and how that the translational work that they've been doing. So with that, it's over to you, Michael, and we're very much looking forward to your talk.
Thank you, Devon, and thank you very much for the invitation. Can you hear me okay? Yeah. Yep.
Yes.
So I'm going to start with a shameless plug for a talk which in a couple of months' time, which some of you will find even more interesting than this one, I think. So this is from Professor Steve Peters at the University of Sheffield, and many of you will know of Steve Peters through his as a psychological support behind the Olympic gold medal winning halls of people like Chris Hoy and Vicky Pendleton at the Beijing and London Olympics, and the 7 world snooker titles of Ronnie O'Sullivan, and his best-selling book, The Chimp Paradox. What you may be less familiar with is his more recent work, which has taken the principles that he used in those kind of performances and put it into effectively an everyday manual that's applicable for pretty much any workplace or life setting, called A Path Through the Jungle. And the work that's been going on in controlled studies of, for example, burnout among nurses in the NHS and the pass rate of army recruits in basic training, that's validated the general applicability of that model. Well, Steve's going to be in Cambridge on the 25th of April, hosted by Jane Warwick at the Faculty of Education and myself, and he's going to talk to us about understanding your mind to reduce stress, optimize performance, and improve interaction with others, and how that can be effective in a workplace and in life in general. So if you, there will be a registration circulating in due course, including from Cambridge Neuroscience. But if you want any further information in the meantime, please contact myself or Jane Warwick at the address shown here. I'll show that again briefly at the end. So programmed axon death is a molecular mechanism now, we think, quite well understood. There may be more to find out yet, but we'll see. And it's been largely understood through the process, through its activation by axon injury, physical injury of axons, transection or crushing of axons. And in that context, in an axon injury, it's known as Wallerian degeneration. But what we now know, and I'll talk you through how we know about this pathway as I go through the talk, but what we now know is not only can this be activated by axon injury, but it can be activated also by both genes and environment. And that is the case both in mouse models of human neurodegenerative disorders and axonal disorders, and quite, as you see there, quite a wide range of some very common and very important neurodegenerative disorders, but also, and this will be the second part of my talk, and this is what really just beginning, is we also are getting clear evidence now of involvement in human disease and not just in animal models. And I will talk through why that matters and why what we know from animal models is not the full story, and why it's really important to look at the human side of that story as well. So, Wallerian degeneration, the strange name comes actually from Augustus Waller, who first described it in 1850. And he observed that when he cut nerves in the tongue of a frog, he saw what he called coagulation and curdling happening in the nerves on the distal side of the axon lesion, the part that's we now know has been separated from the neuronal cell bodies. And he predicted, correctly as it turns out, but with no real way of knowing at the time, that it will be particularly desirable with reference to what he called nervous diseases. Today we would call neurodegenerative disorders, that this will be most relevant. And that, apart from some observations of morphological similarity, that is pretty much where the field stood for the next 139 years, until in 1989, Hugh Perry and Michael Brown at the University of Oxford were working with a particular strain of mouse they thought was just any old wild-type strain of mouse. And they were using Waller's methods, now applied in other organisms, to study peripheral nerve regeneration after a crush of sciatic nerve in the mice. And much to their surprise, instead of seeing the expected Wallerian degeneration of the axons on the distal side of the injury, which they were then hoping to follow up by looking at the process of regeneration, what was affecting it, they found in that particular cohort of mice prolonged survival of the axon on the distal side of the injury. So these became known as the Wallerian degeneration slow mice, WLDS mice, and that was about a 10-fold delay in the degeneration of the axons on the distal side of that injury. And Hugh's response to that at the time was to send a note to Michael Brown, who was in a different department in Oxford at the time, to say, go cut the nerve again. He simply didn't believe initially that this could be a real result. And of course, the most likely interpretation of anything as striking as that is some kind of experimental error. Maybe the nerve wasn't cut properly or something like that. But actually, it turns out this was an entirely reproducible phenotype throughout that breeding colony that was completely absent in other strains of mouse at that time. So they did quite a lot of follow-up work. They showed that this was an intrinsic property of the nerve and not of macrophages infiltrating into that nerve after nerve lesion, and that it was a property of the axons in the nerve and not the glia. And they carried out some basic Mendelian characterization of this phenotype and showed that it was an autosomal dominant phenotype. And then they linked up with Mary Lyon down the road at MLC Harwell near Oxford to start the genetic mapping of that phenotype. But before I tell you about the results of that, my job when I joined Hugh's group as a postdoc was to complete that process and identify the gene. Before I tell you about that, what this phenotype then made possible was the testing of Waller's prediction that this process of Wallerian degeneration was relevant to nervous system disorders and neurodegenerative disorders, because now there was a way to block the process. And the straightforward question to ask then was, can the same genetic change that was present in those mice block axon degeneration in the absence of physical injury? And two ways to study neurodegeneration in the absence of physical injury. One is genetic and the other is toxic, different ways to trigger axon degeneration. And these are two of the early examples that were studied. In Anketo's group in Geneva, in a project that we collaborated on, they crossed a very severe motor axon degeneration phenotype in mice called progressive motor neuronopathy, PMN, which is lethal at about 7 weeks of age in those mice, which characterized with very extensive axon loss very shortly after weaning in the mice. And they showed not only a very dramatic but temporary extension of the survival of those axons after this genetic lesion, but also an extension of the lifespan of those mice. This was the mutation was identified in PMN mice as a loss of function of a tubulin chaperone protein, and which basically means they have a very severe defect in axonal transport. So these axons are being kept alive despite the poor axonal transport that's going on there. Work initially from Jonathan Glass, but reproduced in many groups since, including ours, it was to show that vincristin, a toxin that's actually used as a cancer chemotherapeutic, but happens to be toxic to neurons and axons, and is a dose-limiting, causes dose-limiting side effects in cancer chemotherapy, Vincristine is another drug that will impair microtubules and disrupt microtubules. That again causes axon degeneration, this time in cell culture here. But again, this could be delayed by the presence of the slow Williams degeneration mutation. And this has been reproduced also in vivo in looking at pain studies in vincristine models in animals. So my job in Hugh's group was to lead a small team that eventually identified the gene. And the way we did that was positional cloning. And I think for audiences these days, I probably should run through what that meant, because in the early to mid 90s, this was kind of all the rage. This was what you did to find your disease gene or your interesting phenotype gene. But the world has changed considerably since then with the sequencing of the human genome and mouse genome and many others. So what I'm showing you here, the principles still apply, but the method that you use will be quite different now. So genomes are shuffled during meiosis between generations. So you get this recombination, the crossing over between the maternal and the paternal chromosomes, such that the genetic material in the offspring is shuffled. All chromosomes in the offspring are a combination, apart from the Y, I guess, in the males. All chromosomes there are a combination of genetic material from the grandfather and the grandmother. That none of them is a straightforward inheritance of the entire chromosome. And this creates an opportunity to understand whether the phenotype is segregated with particular molecular markers on that chromosome, like microsatellite markers, for example, that could tell you which part of which chromosome the gene for that phenotype was located on. So you take your family, or in this case, a mouse cross, and track the inheritance of that phenotype through multiple different meiotic events. You localize it down to the region of the chromosome, and in pre-genome sequence days, you then carried out very long but laborious procedures to isolate all the DNA from that part of the chromosome, and then identify the genes within it, and then eventually ideally identify a mutation within one of those genes, focusing, of course, probably on the best candidate genes within that region. Today, you would go direct from here to here using genome sequencers, but you still have to do this critical last step, which is prove that the mutation you have found is causative for that phenotype. And that we did in this case, having identified a candidate mutation at least, was to, the way we did that was to make a transgenic mouse that recapitulated the phenotype of the original slow Williams degeneration mouse. We had found not a missense mutation within a gene, which is the typical kind of mutation you might think of, but a genomic rearrangement of a large chunk of the mouse chromosome here, 85 kilobases, which was triplicated head to tail on mouse chromosome 4, and that had an effect on 3 genes within it. The important one is what you see here is this chimeric gene that's formed across the fusion boundary here. When you follow through to look at what the transcript, the splicing transcript across that boundary predicts in terms of a protein, it predicts a chimeric protein made up of sequences from 2 normal proteins. The pink part is an NAD synthesizing enzyme called NMNAT1, and the blue part here is a fragment of a ubiquitin ligase called UBE4B. And that's the, we took the cDNA for that, we put it into transgenic mice, and we showed that that would recapitulate the phenotype of the original mice. That's the proof that we'd identified the right gene. So the next step was to understand how that's functioning. Is it loss or gain of function? Whereabouts in the cell is it acting? So it turned out the hybrid protein, the chimeric protein, does indeed have NAD synthesis activity. If you take it in as a biochemical isolate and give it the substrates, nicotinamide mononucleotide and ATP, it will make NAD. And that when we and other groups, Jeff Milbrandt and Mark Freeman and others, made enzyme dead versions of that protein, and express that in neurons, those axons did not have a protective phenotype. So that enzyme activity was essential for the phenotype. However, that enzyme activity had to be located in axons. And one of the pieces of evidence for that is shown here, work from Elisabetta Barbetto, when she was a student in my group. If we took simply overexpressing the NMNAT1 protein, the pink part of this protein by itself, overexpressed at reasonably high levels, and then we cut the axons there, we still see fragmentation of the axons in those nerves. But when we took that same protein and deliberately targeted it into axons using a fragment of amyloid precursor protein, which was known to be an axonal targeting sequence, we now regained the protective phenotype. That's one of the pieces of evidence that NAD synthesis, or the NMNAT activity at least, within axons is the important cause of this phenotype. Another piece of evidence a few years later from Laura Conforti and Michela Di Stefano, Laura eventually formed her own group, so still collaborating on this work, was to show that after you injure a mouse static nerve, there is a decrease in the NAD levels after the injury takes place. So there's no starting difference in the basal level of NAD between the wild type nerve and the WRDS nerve. But when you injure those axons, the normal loss of NAD is stabilized by the presence of the WRDS mutation. So that tells you that it is carrying out sustained NAD synthesis after a nerve lesion in a way that the wild type equivalent of it cannot. And importantly for later in the talk, you will also see the increase, a corresponding increase in the substrate of that protein, nicotinamide mononucleotide, and we'll come back to that later, which is also stabilized by the presence of WLDS. And that took about 10 years after identifying the gene before we got to that point. And then we realized that really the critical thing to do is go in the other direction and ask, what is the normal protein that's driving Wallerian degeneration in all of us, if our nerves get injured or if our axons get sick in some other way? And the fairly straightforward hypothesis now in retrospect, but as I say, it took the entire field about 10 years to come up with it, was that if a gain of function of axonal NMNAT is protective for axons, is it a loss of function of axonal NMNAT that is the driver of Wallerian degeneration or programmed axon death normally? And John Gilly in the group was a person who tested that by taking what by then was the 3 known NMNAT isoforms in mammals, 1, 2, and 3, knocking them down in primary neuronal cultures using this microinjection technique that we do, and fluorescently tagging the injected axons at the same time, and simply following them up over the course of the next several days to see whether any of them, in the absence of the corresponding protein or the depletion of that protein, degenerated. And you'll see it's loss of NMNAT2 that is the trigger for axon degeneration in over the course of 1 to 3 days here, without any physical injury taking place. And when WLDS is present, which has that same enzyme activity, the NMNAT activity, those axons are now preserved. So we put forward a model suggesting that WLDS, and it turned out is a much more stable protein than NMNAT2. So the prolonged presence of WLDS after an axon lesion is what enables those axons to survive for longer after a physical injury. John followed that up by looking in vivo at gene trap mice, which have a full loss of function for NMNAT2, a full depletion of the messaging protein. Those mice have a perinatal lethal phenotype, which is caused by the inability to grow long axons. So these are the leg nerves in the late stage embryos of those mice. You can see the truncation of the axon at the knee and the rescue of that axon growth by crossing those animals with WLDS, another piece of evidence that WLDS substitutes for NMNAT2. And the actual cause of death, of course, if they can't grow long axons, is the absence of a phrenic nerve. They don't innervate their diaphragm. And so there's perinatal lethality in those mice. we also showed, although the animals die at birth with the inability to breathe, they are alive in late-stage embryos. So we could take neuronal explants from those late-stage embryos and grow them in cell culture. And both in sympathetic neurons, SCG explants, and dorsal root ganglia, sensory axons, we saw a truncation of axon growth. This is a time course here, initial growth at the normal speed for about 1 millimeter, and then the homozygous nulls kind of hit a brick wall. They can't keep their axons alive without a local supply of that NMT, the NMT2 isoform. That's what happens if you totally take away NMT2. If, on the other hand, you deplete its expression level, which we could do because there is a second gene trap form of those mice where there is a reduction in expression, but not a complete ablation of expression. If we took compound heterozygotes from our mice and the reduced expression mice, we have 30% of normal expression level in those mice. Now, we saw the mice remained healthy overtly, but they did have a deficiency of sensory axons in young animals. And as they became older, they also got a greater deficiency in motor axons as well. And although they were healthy, they're not normal. And one way we showed that was a thermal sensitivity test. So what we do here is we offer the animals a choice of 2 places to stand. One is a plate at a nice comfortable temperature, and the other is a plate that we vary from noxiously warm to noxiously cold. And when the variable plate is also at a comfortable temperature for a mouse, 30 degrees, the animals stand about 50% of the time on each plate. As the plate gets too warm, the wild type animals in grey decrease the amount of time they spend on the plate that's too warm, and correspondingly as it gets too cold. But as you see, the compound heterozygous animals in whatever color that is, brown here, spend longer standing on the plate where it would normally feel uncomfortable. So what we conclude from that provisionally is that the animals can't sense, they don't have a sense that the plate is now warmer than it should be or colder than it should be. Axons from those same animals also have greater sensitivity to vincrestin. This is now a very low dose of vincrestin that is not sufficient to kill wild type axons, lower than the ones I showed earlier in the previous slide where WLDS was protecting. This won't kill wild type axons, but it is sufficient to kill axons which have a depleted level of NMT2. And there is also loss of myelinated axons with age, and there is the animals are starting to fall off a rotor rod too early. So this is a motor phenotype. And to preempt, get you thinking a little bit about what I'll talk about in the later part of the talk, there is a very widespread, at least at the message level, there's a very widespread variation of expression for NMT2 between different people. I'm not sure, this is work from Hui-Chen Liu in Indiana, whether or not there's any correlation with cognition, I think is open to question here, but there is very clearly, and this is also work now produced by Pete Williams and James Tribble's groups in Lund, in using retina, there is very clearly a very wide range of expression level between different people. So the question we need to ask is, if mice with low expression have this greater sensitivity to axonal stressors, what about the people too? So the other part of the mechanism story, the SARM1 protein, comes from collaboration with Mark Freeman and colleagues at that time at the University of Massachusetts in Worcester. So Mark, we sent Mark the mouse cDNA for WLDS quite a while ago now, and Mark put it into flies. They put it into the antenna of a fly and overexpressed it there, and tested after breaking off the antenna of a fly, was there a protective phenotype in flies using the mouse gene? And the answer here is yes. This is the distal part of the axon in those flies surviving after that lesion. Mark then said, this validates the use of Drosophila with all the power of its genetics to understand which other proteins and genes may influence this pathway. And he set about doing chemical mutagenesis in flies to test whether there were other genes that was altering that and screening them. And eventually they found 3 mutations, 3 loss of function mutations in the same gene, which also preserved injured axons. This was a Drosophila ortholog of a protein which in mammals we call SARM1. SARM1 encodes a toll-like receptor adapter protein. That link between innate immunity and axon degeneration is still something that will be intriguing to understand. We don't yet have a full understanding of that, but there's something important in that, I'm sure. What was very clear, however, right from the beginning, is that both in flies and in mice, that taking away SARM1 exactly phenocopied the original WLDS mutation. The work we followed up in our group then was to test whether SARM1 was acting in the same pathway as NMT2. So if we go back to those NMT2 null mice that die at birth, John Gilly crossed those with the SARM1 null mice, and quite startling, this is the most dramatic rescue of that phenotype you could imagine. We go from death at birth to normal lifespan in mice and retaining the full complement of axons right through to old age. And we showed, working together with Richard Riftester from Edinburgh, that they retained the full neuromuscular innervation level into old mice as well. That showed it works on the same pathway, and it really attracted the attention, I think, of Farmer, who then took notice and thought, well, this is a pathway that's worth blocking, and happened to coincide with the discovery of an enzyme activity in SARM1 from Jeff Milbrandt's group, which I'll tell you about in a moment. But the other thing that was evident from that work is that SARM1 must be acting downstream of the loss of NMT2. And one reason we know that is because, unlike WLDS, which would stabilize the NAD and NMN levels in a cut nerve, the removal of SARM1 does not. There is possibly a slight slowing of the loss of NAD, but there is still an ongoing gradual loss of NAD after cutting a SARM1 null nerve. But there is definitely no stabilization of the substrate level, the NMN. In fact, if anything, NMN goes up much higher in a cut SARM1 null nerve than it does in the wild type. So this tells you, this and other evidence shows you that SARM1 is not acting by stabilizing NMT2, it's acting by keeping axons alive despite the loss of NMT2. So this places SARM1 downstream of the loss of NMT2. We also know that WLDS and NMT2 both somehow block the activity of SARM1, but WLDS does it for longer. And as I said, Jeff Milbrandt and Aaron D'Antonio around that time identified an intrinsic NA Dase activity in SARM1. So SARM1 actually degrades NAD if you give it those substrates, NAD as a substrate in a biochemical assay. The next step for us then was to ask, how does the loss of NMT2 talk? What does it do to SARM1 to make those axons degenerate? So we were studying the first sign of that something interesting was going on here was the experiments we were doing in primary neuronal cultures to ask if we could cause axon degeneration purely by depleting NAD. And we were doing that with an inhibitor of the rate-limiting step in that pathway, catalyzed by an enzyme called NAMT, and the inhibitor is FK866. And as you see here, giving FK866 to cultures, far from killing them, actually preserved axons after an injury. It was doing effectively phenocopying WLDS. This causes a little bit of confusion at first, but the best hypothesis we could come up with was that the intermediate in that pathway, called nicotinamide mononucleotide, the substrate of NMT2, is a toxic intermediate. We tested that by making transgenic mice and cell culture work using a bacterial enzyme called NMN deamidase. NMN deamidase depletes NMN by converting it into a different metabolite, nicotinic acid mononucleotide. And when you do that, the axons are protected. And they are actually protected as strongly as WLDS mice and as SARM1 null mice. And all we've done here is remove NMN. So the explanation for that became clear when a group of Yongwan Zhao in Beijing showed that NMN is an activator of the SARM1 NA Dase. So SARM1, as I said on the previous slide, degrades NAD in a biochemical assay, but if you do that in the presence of NMN, it does so faster. So the situation we have is that NMT2 is an inhibitor of SARM1, and in a healthy axon, it keeps SARM1 quiet and keeps the NAD levels normal. If you injure the axon, or there is a disease which depletes NMT2, NAD levels fall because you lose the synthetic enzyme. But in addition, the substrate, nicotinamide mononucleotide, goes up. And this activates SARM1, which is an NADase, so you get a second hit to the NAD levels. Whether the decrease of NAD is the thing that actually kills the axon, or whether there's something else, that's still open to question in my mind. SARM1 does have other enzyme activities, which I don't have time to go into. And even if it's working through NAD, there's lots of different ways that a loss of NAD could kill an axon. But it's certainly one of the best candidates for what may be going on here. It's slightly more complicated than that, because NAD is also able to block the activation by NMN if there's enough of it there. So the physiological concentration of NAD in an axon, as far as we know, is about 200 micromolar, which is around about the peak of these curves here. So even at NAD levels close to physiological, there is a leveling off of the rate of activity of the enzyme as you increase NAD. So this suggests NAD is starting to block the activation of SARM1 by nicotinamide mononucleotide. But basically close to physiological levels on both nucleotides, NMN is promoting the activity of SARM1. NAD is decreasing, is opposing that action. So a summary so far is that programmed axon death can be activated by both injury and disease, that both genes, SARM1 and NMT2, are affected by both genes and environment that can activate this pathway, that NAD metabolism is very closely connected with that axon survival, and it's loss of NMT2 that leads to SARM1 activation, which in turn kills axons by a mechanism that is still not completely understood. SARM1 is activated by NMN, the substrate of NMT2, which binds in competition with NAD, and it's a gain of axonal NMT and loss of SARM1 that are both protective. Go back. And that protection can be permanent in the case of the double knockouts, as NMT2 SARM1 double knockouts, if the pathway is activated specifically enough. So we've gone from that 2 to 3 week extended survival in the context of a transected axon at the beginning, now to potentially lifelong rescue when that pathway is very specifically activated. And that's where the talk would end if it were not for this guy, George Box, a British statistician, I think in around about the 1950s, who told us that all models are wrong, but some are useful. And that, I think, is a very pertinent quote here, because we need to ask, in those animal and cell culture models that I've described so far, what's useful and what's wrong or potentially wrong? I'd say that what is useful is it's told us beyond any doubt that this pathway of programmed axon death is relevant to non-injury diseases. There's just simply too many animal models that do not involve physical injury for that not to be the case. But does it tell us which human diseases? I would say no, or at least it would be premature to make that conclusion, because there are so many limitations of animal models. And I'll just run through on this slide what some of those limitations are. So the obvious ones we think about all the time, I think, are differences in protein coding, sequencers, splicing, gene expression, differences in anatomy, And in the context of axons, particularly axon length, so a mouse's axons are approximately 1/20th of the length of ours. And difference in longevity, of course. But there's a whole lot of others that we don't think about so much, and in my view, need to think about much more. Mice are inbred, or the mice we use are generally inbred. Humans are genetically diverse. I showed earlier that huge range of NMT2 expression levels in humans. If we use black 6 mice in an animal house, they've all got the same, pretty much the same NMT2 expression level. And we need to incorporate that somehow into our models, or at least into our thinking. Mice also have, the way we keep them, have a very highly controlled environment, a 12-hour light dark cycle, uniform temperature, a 24/7, 365. I mean, it's really not remotely the environment we live in. They're not exposed to viruses. They're not exposed to changes in diet. They don't drink alcohol. There's so many differences between the way they live and the way we live. And to my mind, that is not modeling real human disease. And more importantly, the models we use, what we do is we tend to hit one risk factor very hard. We overexpress a mutant transgene, or we give a very heavy dose of toxin, whereas most human disease is multifactorial, involving multiple genes and environment. So by now, you should really be asking the question, do mice tell us anything at all that's relevant to real world human disease? And it might sound a very provocative question, but I think it's a very important one to ask. Before you all run away in despair, the answer is yes, but they don't tell us the whole story. So the mice I've already told you about, the NMT2 nulls and the compound heterozygous with low NMT2 expression level, they have direct counterparts in rare human disorders. Unfortunately, there are some very rare cases of stillbirth in humans with a full compound heterozygous null mutations in both NMT2 alleles. We worked together with a group in Cleveland to show that those have a full loss of function, both of those alleles, and they cause this gross phenotype with birth with no, or stillbirth with no skeletal muscle and brain defects on MRI. There are also hypermorphic alleles in humans, so partial loss of function, which are associated with childhood onset peripheral neuropathy or polyneuropathy, or a neuropathic pain disorder, including in nerve biopsies with the deficiency of sensory axons in the distal limb. So there seem to be very rare biallelic mutations in MT2, which correspond, broadly speaking, with the mouse phenotypes that we saw. So very likely causative. When we turn to SARM1, and in particular in the context of motor neuron disease, it is a little bit more misleading what I think the mice told us. So first of all, the encouraging news, progressive motor neuronopathy, that phenotype I showed earlier that was very dramatically but temporarily rescued by the presence of WLDS. What we now know is that does have it, that mutations in that gene do have a direct counterpart again in a rare human disorder, and that's progressive encephalopathy with distal SMA. This is a very rare disorder in that case, I think some families in Italy. And again, it is likely that what is happening in those individuals is broadly related to what happened in the PMN mice, which were able to rescue or temporarily rescue with that phenotype. However, when that study was first published in 2003, there was a lot of excitement in the ALS community that it might be possible to rescue what at that time was, and still is to some degree, the most widely used model of ALS in mice, which is the transgenic mutations of SOD1. Unfortunately, both for WLDS and for SARM1 knockout, that has not been the case. This is work from Bob Brown's group. There was an earlier study from Jonathan Glass looking at SOD1 with WLDS. There is no protection either in the morphology of the axons or in the phenotype in the symptoms of the animals. And that for a very long time seemed to be the end of the story. And it seemed that, you know, perhaps in ALS, this wasn't so important after all. However, human genetics seems to be telling us a very different story. First of all, work from King's College London and other groups shows that there is a GWAS association with the SARM1 locus on chromosome 17 and sporadic ALS. And this is one of the strongest genome GWAS associations in the genome for sporadic ALS. That does not by itself prove that it's SARM1 gene that's causative in that region, but something is. But the lead SNP for that does map directly within a SARM1 intron. So it's one of, not only is it a good candidate, it's extraordinarily close to the polymorphism that's driving that, or that's marking it at least. On the back of that, we decided to have a look in the Project Mine whole genome sequence database at whether SARM1 coding sequences, even if they're rare alleles, were doing anything interesting. And what we found was that a domain of SARM1 that is inhibitory, called the ARM domain, had a very high prevalence of patient-specific mutations in the Project Mine database, whereas that same region was completely devoid of any control-specific mutations. So Project Mine has, I think, about 4,600 ALS cases, full sequence, and about 1,800 controls. You would expect possibly more patient variants than controls on the whole because there's more patients in there, but you would not expect zero in the controls and all of these in the mutant in the patients. Disruption of an inhibitory domain, we figured, could cause a gain of function of the protein, which we would expect to be harmful because it's a pro-degenerative protein. So we tested that. Again, this was John Gillies' work. He showed that approximately half of those variants in an NADase assay with a biochemical protein assay could catalyze the degradation of NAD approximately 20 times faster than the wild type enzyme. This is a huge increase in the rate of degeneration, rate of NAD turnover in that enzyme, and it raised a lot of questions about why these people survive even at all with, you know, a late onset of ALS. Is a relatively late onset disorder in all of those particular patients. I think the average age of onset was around about 60 in there. But they do eventually have degeneration. And there was an association, a very strong association of these still rare alleles in ALS with patients, 13 cases in the patients and 0 in controls. And similar data in other cohorts. We didn't have enough patients overall to do like an independent replication. That is a limitation of that study, but there is certainly a very strong association in the cohorts we studied of SARM1 gain of function with the presence of sporadic ALS. We've also found that not only can genetics activate SARM1, but so can environmental toxins. So Andrea Loreto in the group has been studying a compound called Vaco. Vaco is a nicotinamide analog, which used to be used as a rat poison until about the 1970s when it was banned. And it was banned because it had been used in suicides, and individuals who survived the suicide attempt invariably developed a widespread neurological phenotype. As you see here, widespread involvement of autonomic, peripheral, and central nervous system function developed within hours of digestion of, this is the old name of Vaco. So it was a very lethal neurotoxin, not only in rats, but also in people. And we had reason to believe from a paper that was published several years ago that it could be talking to this pathway. So we tested it. Again, in cell culture, we gave it to cell cultures of mouse neurons. It was absolutely lethal to both the axons and the neuronal cell bodies. But when we tested the SARM1 knockout, they were completely oblivious to the presence of the Vaco, no matter how much Andrea added to those cultures. They just kept on growing. So this was an absolutely, again, very specific activation of the pathway, this time by an environmental toxin. The reason for that was eventually identified. Like nicotinamide mononucleotide, the natural activator of SARM1, a metabolite of Vaco, VMN, but also binds to exactly the same site in the ARM domain and activates the protein. So we've got a pretty full explanation of what's going on there and why Vaco is so lethal and why it's acting through SARM1. There is a second known environmental activator of SARM1 or an environmental factor that's causing some kind of SARM1 dependent axon death, and those are a number of viruses. So far, we only know of 3 or 4, West Nile virus, La Crosse virus, rabies, and Zika. They are all talking to SARM1 in some way, either upregulating it or doing something, possibly activating it, but they're all causing SARM1 dependent axonal death. This raises a very interesting question of, did SARM1 first evolve as some type of host defense against viruses? If you could lose an axon very quickly, then you could stop a virus from spreading through axonal transport. So this was collaborative work we did with Julia Edgar's group, and Julia made these great videos here showing if you didn't have SARM1, a virus could spread by axonal transport, replicate in the cell body, and spread to the next cell. But if you do have SARM1, the virus would come along, SARM1 would kill the distal part of the axon in response to the virus and stop the virus from spreading. And then in peripheral nerve, at least, you could regenerate the distal end of the axon. That's still a hypothesis, but I think it's an extremely interesting one to test. We'd love to know which other viruses talk to this pathway, and crucially, how they talk to the pathway. Work from that Christina Antonio did in my group in collaboration with the Crawford and Edgar study here, showed that while Zika is also causing a decrease in NAD level in the Zika treated cultures, the presence or absence of SARM1, the difference between this group and this group, was making no further difference. So whatever Zika is doing, it's not, whatever someone is doing here, it's not an activation of the NADase that's causing the degeneration. There's a lot more mysteries and interesting things about this protein yet to be discovered, I think. The other context in human disease that has come up in recent years is a function of SARM1, not only in axon degeneration, but also in retinal disorders in the photoreceptor neurons. And these don't actually have proper axons. These are effectively cell bodies. And what is happening here is a loss of the other NMDT in neurons, NMDT1. This is a nuclear protein, but when there is a loss of function mutation in that, in a rare disorder called LCA9, Leber's congenital amaurosis, there is a loss of the NMDT activity, and there is an activation, at least in animal models, of SARM1, synthesizing its product, cyclic ADP ribose. And if you take away SARM1, you can totally rescue those animal models. So this again suggests that there is a wider involvement of SARM1 in neurodegenerative disease, not just in axonal disorders. Which leads to the important question of, where does the axon specificity come from? If you activate SARM1 in any neuronal compartment, it will kill that compartment. And if you activate it with genetic mutation, gain of function mutation, or with a toxin like Vaco, you will activate it in the cell bodies as well as the axon, and both the cell bodies and the axons will die. And also, if you take away the soma NMDT, the NMDT1, again, the cell bodies will die. So it's the loss of NMDT2 that provides the axon specificity in this situation, not the SARM1. And finally, what the direction that we're heading right now is very much about the human genetics of this pathway, and especially also SARM1 loss of function variants in the human population. So work from Stacey Gold a couple of years ago in the group showed that the mouse heterozygous null for SARM1 had a partial protective phenotype, both in axotomy and in vincristine assays. And this was not as strongly protected as homozygous null, but still was significantly different from wild type. Melinda Ademi, a student who's just submitted a thesis, has gone on from that to look in the human population in the NOMAD database. What are the human variants that might induce a loss of function of SARM1? Again, using cell culture and enzyme activity assays, she's shown that there's quite a range of different variants that are unable to cause axon degeneration, natural variants in the human population, and 2 of them actually cause a dominant negative phenotype. So even the inheritance of a single allele would very strongly block axon degeneration in those individuals. And this leads on to this kind of situation that we're proposing exists in the human population, that there is a range of vulnerability for axons in the human population to other types of axonal stress. And there will be a hand, we don't know for sure the shape of this curve, of course, but it's likely there'll be a handful of people at each extreme of that, and most of us are in the middle here. But the important areas will be here when there's significant increase or decrease of disease risk. We know there are NMT2 null alleles, those stillbirth cases, which are non-viable. We know there are lower NMT2, the hypermorph people with erythromelalgia, and SARM1 gain of function variants with a risk factor for ALS. We know there is heterozygous loss of function of SARM1, the work I've just shown you from Melinda, which is protective. And we know there is a handful of dominant negative alleles, which are likely to be more strongly protective. If that is the case, this suggests that we can use what we might call a natural clinical trial. We can take loss of function variants for SARM1, which are effectively doing what drug companies would love to do, with blocking SARM1 with a drug. And we can ask the question, are they underrepresented in specific neurodegenerative diseases? And among the best candidates for that would be the ones where there has been protection in the corresponding mouse models: diabetic neuropathy, Parkinson's disease, multiple sclerosis, EAE at least, glaucoma, and also Alzheimer's disease, which we've effectively been unable to model effectively in mice. So that's an open question still in humans, but clearly a very important one. So that's one of the directions we are heading right now. So in summary of part 2, animal models are a guide to what's happening in human disease, but they only tell us part of the story. Biallelic NMT2 loss of function is a cause of very severe axonal degeneration or growth failure in mice, and has corresponding human phenotypes. SARM1 gain of function is significantly associated with sporadic ALS, and SARM1 can also be activated by environmental factors such as toxins and some viruses. There appears to be involvement in retinal disorders as well, and loss of function alleles provide an opportunity to test in which diseases it will be useful to block this pathway. So finally, thanks to everyone involved. Huge part of the work I've explained has been from John Gilley on the NMT2 work, also supported by Andrea Loretto, Melinda Ademi, as work I've shown, and Christina I've also shown. There's a lot of other people in the group whose work I haven't had time to talk about, but obviously doing some very interesting work going forward, and the major collaborators on this part of the work.
Thank you, Michael, for a wonderful insight into your work. Join us next time when we welcome Dr. Michael Spitale. Michael is currently a postdoctoral researcher at the Effective Intelligence and Robotics Laboratory at the Department of Computer Science and Technology here at the University of Cambridge. She is supervised by Professor Hatice Gunes. Her research activities are grounded in the social robotics area. She has a background in effective computing, child-robot interaction, and machine learning applications to human behavioral analysis. She will speak to us about children-agent interaction for assessment and rehabilitation, from linguistic skills to mental well-being. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below. See you next time.